Phenotypic plasticity. Phenotypic plasticity refers to some of the changes in an organism's behavior, morphology and physiology in response to a unique environment. 1. The way in which organisms cope with environmental variation. Phenotypic plasticity encompasses all types of environmentally induced changes, e.g. morphological, physiological, behavioral, phenological, that may or may not be permanent throughout an individual's lifespan. 3. The term used to describe developmental effects on morphological characters, but is now more broadly used to describe all phenotypic responses to environmental change, such as acclimatization, as well as learning. The special case when differences in environment induce discrete phenotypes is termed polyphenism. Phenotypic plasticity is the ability of one genotype to produce more than one phenotype when exposed to different environments. Each line here represents a genotype. Horizontal lines show that the phenotype is the same in different environments. Slanted lines show that there are different phenotypes in different environments, and thus indicate plasticity. Plasticity is more important for immobile organisms, e.g. plants, than mobile organisms, e.g. most animals, as mobile organisms can often move away from unfavorable environments. 4. Nevertheless, mobile organisms also have at least some degree of plasticity in at least some aspects of the phenotype. 2. One mobile organism with substantial phenotypic plasticity is a Cyrthosyphon of the aphid family, which exhibits the ability to interchange between asexual and sexual reproduction, as well as growing wings between generations when plants become too populated. 5. Fleas, Daphnia magna, have shown both phenotypic plasticity and the ability to genetically evolve to deal with the heat stress of warmer, urban pond waters. 2. Examples. Plants. Phenotypic plasticity of sorghum flowering time evaluated from 7 environments. The identified phenotypic plasticity is more important for immobile organisms, e.g. plants, than mobile organisms, e.g. most animals, as mobile organisms can often move away from unfavorable environments. 4. Nevertheless, mobile organisms also have at least some degree of plasticity in at least some aspects of the phenotype. 2. One mobile organism with substantial phenotypic plasticity is a Cyrthosyphon of the aphid family, which exhibits the ability to interchange between asexual and sexual reproduction, as well as growing wings between generations when plants become too populated. 5. Water fleas, Daphnia magna, have shown both phenotypic plasticity and the ability to genetically evolve to deal with the heat stress of warmer, urban pond waters. 2. Examples. Plants. Phenotypic plasticity of sorghum flowering time evaluated from 7 environments. The photothermal time, a performance-independent index, quantifies the relevant environmental input and enables a systematic framework for modeling, explaining, and predicting phenotypic values under natural conditions. 6. In plants includes the timing of transition from vegetative to reproductive growth stage, the allocation of more resources to the roots in soils that contain low concentrations of nutrients, the size of the seeds an individual produces depending on the environment. 7. And the alteration of leaf shape, size, and thickness. Leaves are particularly plastic, and their growth may be altered by light levels. Leaves tend to be thicker, which maximizes photosynthesis in direct light, and have a smaller area, which cools the leaf more rapidly due to a thinner boundary layer. Conversely, leaves grown in the shade tend to be thinner, with a greater surface area to capture more of the limited light. 9th 10th dandelion are well known for exhibiting considerable plasticity in form when growing in sunny versus shaded environments. Transport proteins present in roots also change depending on the concentration of the nutrient and the salinity of the soil. 11th Some plants, Mesembryanthemum crystallinum for example, are able to alter their photosynthetic pathways to use less water when they become water or salt stressed. 12th Phenotypic plasticity. It is hard to explain and predict the traits when plants are grown in natural conditions unless an explicit environment index can be obtained to quantify environments. Identification of such explicit environment indices from critical growth periods being highly correlated with sorghum and rice flowering time enables such predictions. Additional work is being done to support the agricultural industry, which faces severe challenges in prediction of crop phenotypic expression in changing environments. Since many crops supporting the global food supply are grown in a wide variety of environments, understanding and ability to predict crop genotype by environment interaction will be essential for future food stability. 14. Phytohormones and leaf plasticity. Leaves are very important to a plant in that they create an avenue where photosynthesis and thermoregulation can occur. Evolutionarily, the environmental contribution to leaf shape allowed for a myriad of different types of leaves to be created. 15. Leaf shape can be determined by both genetics and the environment. 16. Environmental factors, such as light and humidity, have been shown to affect leaf morphology. 17. Giving rise to the question of how this shape change is controlled at the molecular level. This means that different leaves could have the same gene but present a different form based on environmental factors. Plants are sessile, so this phenotypic plasticity allows the plant to take in information from its environment and respond without changing its location. In order to understand how leaf morphology works, the anatomy of a leaf must be understood. The main part of the leaf, the blade or lamina, consists of the epidermis, mesophyll, and vascular tissue. The epidermis contains stomata which allows for gas exchange and controls perspiration of the plant. The mesophyll contains most of the chloroplast where photosynthesis can occur. Developing a wide blade lamina can maximize the amount of light hitting the leaf, thereby increasing photosynthesis. However, too much sunlight can damage the plant. Wide lamina can also catch wind easily, which can cause stress to the plant, so finding a happy medium is imperative to the plant's fitness. The genetic regulatory network is responsible for creating this phenotypic plasticity and involves a variety of genes and proteins regulating leaf morphology. Phytohormones have been shown to play a key role in signaling throughout the plant, and changes in concentration of the phytohormones can cause a change in development. 18. Studies on the aquatic species Ludwigia arquata have been done to look at the role of abscisic acid (ABA), as L. arquata is known to exhibit phenotypic plasticity and has two different types of leaves, the aerial type leaves that touch the air and the submerged type leaves that are underwater. 19. Underwater shoots of L. arquata, the plant was able to produce aerial type leaves underwater, suggesting that increased concentrations of ABA in the shoots, likely caused by air contact or a lack of water, triggers the change from the submerged type of leaf to the aerial type. This suggests ABA's role in leaf phenotypic change and its importance in regulating stress through environmental change, such as adapting from being underwater to above water. In the same study, another phytohormone, ethylene, was shown to induce the submerged leaf phenotype unlike ABA, which induced aerial leaf phenotype. Because ethylene is a gas, it tends to stay endogenously within the plant when underwater. This growth in concentration of ethylene induces a change from aerial to submerged leaves and has also been shown to inhibit ABA production, further increasing the growth of submerged type leaves. These factors, temperature, water availability, and phytohormones, contribute to changes in leaf morphology throughout a plant's lifetime and are vital to maximize plant fitness. Animals. The developmental effects of nutrition and temperature have been demonstrated. 20. The gray wolf, Canis lupus, has wide phenotypic plasticity. 22. Additionally, male speckled wood butterflies have two morphs, one with 3 dots on its hindwing, and one with 4 dots on its hindwings. Development of the fourth dot is dependent on environmental conditions, more specifically, location and the time of year. 23. Amphibians, Pristomantis mutabilis has remarkable phenotypic plasticity. 24. As well as Agallichnus callidrias, whose embryos exhibit phenotypic plasticity, hatching early in response to disturbance to protect themselves. Another example is the southern rockhopper penguin. 25. Rockhopper penguins are present at a variety of climates and locations: Amsterdam Island's subtropical waters, Kerguelen Archipelago and Crozet Archipelago's subantarctic coastal waters. 5. Due to the species plasticity, they are able to express different strategies and foraging behaviors depending on the climate and environment. 25. A main factor that has influenced the species' behavior is where food is located. 25. Temperature. Responses to temperature are essential among ectothermic organisms, as all aspects of their physiology are directly dependent on their thermal environment. Such thermal acclimation entails phenotypic adjustments that are found commonly across taxa, such as changes in the lipid composition of cell membranes. Temperature change influences the fluidity of cell membranes by affecting the motion of the fatty acyl chains of glycerophospholipids. Because membrane fluidity is critical for cell function, ectotherms adjust the phospholipid composition of their cell membranes such that the strength of van der Waals forces within the membrane is changed, thereby maintaining fluidity across temperatures. 26. Diet. Plasticity of the digestive system allows some animals to respond to changes in dietary nutrient composition. 27, 28, diet quality, 29, 30, and energy requirements. 31, 32, 33. In the nutrient composition of the diet, the proportion of lipids, proteins and carbohydrates may occur during development, e.g. weaning, or with seasonal changes in the abundance of different food types. These changes can elicit plasticity in the activity of particular digestive enzymes on the brush border of the small intestine. For a few days after hatching, nestling house sparrows Passer domesticus transition from an insect diet, high in protein and lipids, to a seed-based diet that contains mostly carbohydrates. This diet change is accompanied by twofold increase in the activity of the enzyme maltase, which digests carbohydrates. 27. Acclimatizing animals to high-protein diets can increase the activity of aminopeptidase N, which digests proteins. 28, 34. Poor quality diets, those that contain a large amount of non-digestible material, have lower concentrations of nutrients, so animals must process a greater total volume of poor quality food to extract the same amount of energy as they would from a high-quality diet. Many species have quality diets by increasing their food intake, enlarging digestive organs, and increasing the capacity of the digestive tract, e.g. prairie voles. 33. Mongolian gerbils, 30. Japanese quail, 29. Wood ducks, 35. Mallards, 36. Poor quality diets also result in lower concentrations of nutrients in the lumen of the intestine, which can cause a decrease in the activity of several digestive enzymes. 30. When consume more food during periods of high energy demand, e.g. lactation or cold exposure in endotherms, this is facilitated by an increase in digestive organ size and capacity, which is similar to the phenotype produced by poor quality diets. Lactation, common degus, Octodon degus, increase the mass of their liver, small intestine, large intestine and cecum by 15–35%. 31. Increase in food intake do not cause changes in the activity of digestive enzymes because nutrient concentrations in the intestinal lumen are determined by food quality and remain unaffected. 31. Intereating also represents a temporal increase in food intake and can induce dramatic changes in the size of the gut. the Burmese python Python Melurus bivitidis can triple the size of its small intestine just a few days after feeding. 37. AMY2B, alpha amylase 2B, is a gene that codes a protein that assists with the first step in the digestion of dietary starch and glycogen. An expansion of this gene in dogs would enable early dogs to exploit a starch-rich diet as they fed on refuse from agriculture. That the wolves and dingo had just 2 copies of the gene, and the Siberian husky that is associated with hunter-gatherers had just 3 or 4 copies, whereas the Saluki that is associated with the Fertile Crescent where agriculture originated had 29 copies. The results show that on average, modern dogs have a high copy number of the gene, whereas wolves and dingoes do not. The number of AMY2B variants likely already existed as a standing variation in early domestic dogs, but expanded more recently with the development of large agriculturally based civilizations. 38. Parasitism. Infection with parasites can induce phenotypic plasticity as a means to compensate for the detrimental effects caused by parasitism. Invertebrates respond to parasitic castration or increased parasite virulence with fecundity compensation in order to increase their reproductive output, or fitness. For example, water fleas Daphnia magna, exposed to microsporidian parasites, produce more offspring in the early stages of exposure to compensate for future loss of reproductive success. 39. A reduction in fecundity may also occur as a means of redirecting nutrients to an immune response, 40, or to increase longevity of the host. 41. The particular form of plasticity has been shown in certain cases to be mediated by host-derived molecules, e.g. schistosomin in snails Limnia stagnalis infected with trematodes Trichobilharzia ocellata, that interfere with the action of reproductive hormones on their target organs. 42. Reproductive effort during infection is also thought to be a less costly alternative to mounting resistance or defense against invading parasites, although it can occur in concert with a defense response. 43. Hosts can also respond to parasitism through plasticity in physiology aside from reproduction. House mice infected with intestinal nematodes experience decreased rates of glucose transport in the intestine. To compensate for this, mice increase the total mass of mucosal cells responsible for glucose transport, in the intestine. This allows infected mice to maintain the same capacity for glucose uptake and body size as uninfected mice. 44. Phenotypic plasticity can also be observed as changes in behavior. In response to infection, both vertebrates and invertebrates practice self-medication, which can be considered a form of adaptive plasticity. 45. Various species of non-human primates infected with intestinal worms engage in leaf swallowing, in which they ingest rough, whole leaves that physically dislodge parasites from the intestine. The leaves irritate the gastric mucosa, which promotes the secretion of gastric acid and increases gut motility, effectively flushing parasites from the system. 4. The term, self-induced adaptive plasticity, has been used to describe situations in which a behavior under selection causes changes in subordinate traits that in turn enhance the ability of the organism to perform the behavior. 47. Birds that engage in altitudinal migration might make trial runs, lasting a few hours that would induce physiological changes that would improve their ability to function at high altitude. 47. Caterpillars Gramia interrupta infected with tachnid flies increase their survival by ingesting plants containing toxins known as pyrrolizidine alkaloids. The physiological basis for this change in behavior is unknown. However, it is possible that, when activated, the immune system sends signals to the taste system that trigger plasticity in feeding responses during infection. 45. Reproduction. Hatch rates for red-eyed tree frog tadpoles depends on predation. The red-eyed tree frog, Agalaichnus call-idrias, is an arboreal frog hylid that resides in the tropics of Central America. Unlike many frogs, the red-eyed tree frog has arboreal eggs which are laid on leaves hanging over ponds or large petals and, upon hatching, the tadpoles fall into the water below. One of the most common predators encountered by these arboreal eggs is the cat-eyed snake, Leptodira septentrionalis. In order to escape, red-eyed tree frogs have developed a form of adaptive plasticity, which can also be considered phenotypic plasticity. When it comes to hatching age, the clutch is able to hatch prematurely and survive outside of the egg 5 days after oviposition when faced with an immediate threat of predation. The egg clutches take in important information from the vibrations felt around them and use it to determine whether or not they are at risk of predation. In the event of a snake attack, the clutch identifies the threat by the vibrations given off which, in turn, stimulates hatching almost instantaneously. Experiment conducted by Karen Warkentin, hatching rate and ages of red-eyed tree frogs were observed in clutches that were and were not attacked by the cat-eyed snake. When a clutch was attacked at 6 days of age, the entire clutch hatched at the same time, almost instantaneously. However, when a clutch is not presented with the threat of predation, the eggs hatch gradually over time with the first few hatching around 7 days after oviposition, and the last of the clutch hatching around day 10. Karen Warkentin's study further explores the benefits and trade-offs of hatching plasticity in the red-eyed tree frog. 48. Evolution. It is thought to be an evolutionary adaptation to environmental variations that is reasonably predictable and occurs within the lifespan of an individual organism, as it allows individuals to fit their phenotype to different environments. 49-50. If the optimal phenotype in a given environment changes with environmental conditions, then the ability of individuals to express different traits should be advantageous and thus selected for. Hence, phenotypic plasticity can evolve if Darwinian fitness is increased by changing phenotype. 52. A similar logic should apply in artificial evolution attempting to introduce phenotypic plasticity to artificial agents. 53. However, the fitness benefits of plasticity can be limited by the energetic costs of plastic responses, e.g. synthesizing new proteins, adjusting expression ratio of isozyme variants, maintaining sensory machinery to detect changes, as well as the predictability and reliability of environmental cues. 54. See beneficial acclimation hypothesis. Freshwater snails, Visivergata, provide an example of when phenotypic plasticity can be either adaptive or maladaptive. In the presence of a predator, bluegill sunfish, these snails make their shell shape more rotund and reduce growth. This makes them more crush-resistant and better protected from predation. However, these snails cannot tell the difference in chemical cues between the predatory and non-predatory sunfish. The snails respond inappropriately to non-predatory sunfish by producing an altered shell shape and reducing growth. These changes, in the absence of a predator, make the snails susceptible to other predators and limit fecundity. Therefore, these freshwater snails produce either an adaptive or maladaptive response to the environmental cue depending on whether predatory sunfish are present or not. 55-56. Given the practical importance of temperature and its predictable variability over large spatial and temporal scales, adaptation to thermal variation has been hypothesized to be a key mechanism dictating the capacity of organisms for phenotypic plasticity. 50. The magnitude of thermal variation is thought to be directly proportional to plastic capacity, such that species that have evolved in the warm, constant climate of the tropics have a lower capacity for plasticity compared to those living in variable temperate habitats. Termed the climatic variability hypothesis, this idea has been supported by several studies of plastic capacity across latitude in both plants and animals. 59. However, recent studies of Drosophila species have failed to detect a clear pattern of plasticity over latitudinal gradients, suggesting this hypothesis may not hold true across all taxa or for all traits. 60. Some researchers propose that direct measures of environmental variability, using factors such as precipitation, are better predictors of phenotypic plasticity than latitude alone. 61. Selection experiments and experimental evolution approaches have shown that plasticity is a trait that can evolve when under direct selection and also as a correlated response to selection on the average values of particular traits. 62. Temporal plasticity. Temporal plasticity, also known as fine-grained environmental adaptation, 63, is a type of phenotypic plasticity that involves the phenotypic change of organisms in response to changes in the environment over time. Animals can respond to short-term environmental changes with physiological, reversible, and behavioral changes. Plants, which are sedentary, respond to short-term environmental changes with both physiological and developmental, non-reversible changes. Animals can respond to short-term environmental changes with physiological, reversible, and behavioral changes. Plants, which are sedentary, respond to short-term environmental changes with both physiological and developmental, non-reversible changes. 64. Temporal plasticity takes place over a time scale of minutes, days, or seasons, and in environments that are both variable and predictable within the lifespan of an individual. Temporal plasticity is considered adaptive if the phenotypic response results in increased fitness. 65. Reversible phenotypic changes can be observed in metameric organisms such as plants that depend on the environmental conditions each metamer was developed under. 60. Under some circumstances, early exposure to specific stressors can affect how an individual plant is capable of responding to future environmental changes. Metaplasticity. 66. Plasticity and climate change. Unprecedented rates of climate change are predicted to occur over the next 100 years as a result of human activity. 67. Phenotypic plasticity is a key mechanism with which organisms can cope with a changing climate, as it allows individuals to respond to change within their lifetime. 68. Is thought to be particularly important for species with long generation times, as evolutionary responses via natural selection may not produce change fast enough to mitigate the effects of a warmer climate. The North American red squirrel, Tamiocyrus ebsenicus, has experienced an increase in average temperature over this last decade of almost 2 degrees Celsius. This increase in temperature has caused an increase in abundance of white spruce cones, the main food source for winter and spring reproduction. In response, the mean lifetime parturition date of this species has advanced by 18 days. Food abundance showed a significant effect on the breeding date with individual females, indicating a high amount of phenotypic plasticity in this trait. 6. Molecular evolution. Evolution describes how inherited DNA and/or RNA change over evolutionary time, and the consequences of this for proteins and other components of cells and organisms. Molecular evolution is the basis of phylogenetic approaches to describing the tree of life. Molecular evolution overlaps with population genetics, especially on shorter timescales. Molecular evolution include the origins of new genes, the genetic nature of complex traits, the genetic basis of adaptation and speciation, the evolution of development, and patterns and processes underlying genomic changes during evolution. History. The theory of molecular evolution starts in the early 20th century with comparative biochemistry, and the use of fingerprinting, methods such as immunoassays, gel electrophoresis, and paper chromatography in the 1950s to explore homologous proteins. 1. The advent of protein sequencing allowed molecular biologists to create phylogenies based on sequence comparison, and to use the differences between homologous sequences as a molecular clock to estimate the time since the most recent common ancestor. 1. The surprisingly large amount of molecular divergence within and between species inspired the neutral theory of molecular evolution in the late 1960s. 4. 5. 6. Neutral theory also provided a theoretical basis for the molecular clock, although this is not needed for the clock's validity. After the 1970s, nucleic acid sequencing allowed molecular evolution to reach beyond proteins to highly conserved ribosomal RNA sequences, the foundation of a reconceptualization of the early history of life. 1. The Society for Molecular Biology and Evolution was founded in 1982. Molecular phylogenetics. Multiple sequence alignment, in this case DNA sequences, and illustrations of the use of substitution models to make evolutionary inferences. The data in this alignment, in this case a toy example with 18 sites, is converted to a set of site patterns. The site patterns are shown along with the number of times they occur in alignment. These site patterns are used to calculate the likelihood given the substitution model in a phylogenetic tree, in this case an unrooted 4-taxon tree. It is also necessary to assume a substitution model to estimate evolutionary distances for pairs of sequences. Distances are the number of substitutions that have occurred since sequences had a common ancestor. Evolutionary distance equation, D12, is based on the simple model proposed by Jukes and Cantor in 1969. The equation transforms the proportion of nucleotide differences between taxa 1 and 2, P12 equals 4/18. The 4 site patterns that differ between taxa 1 and 2 are indicated with asterisks into an evolutionary distance, in this case D12 equals 0.2635 substitutions per site. Molecular phylogenetics uses DNA, RNA, or protein sequences to resolve questions in systematics, i.e. about their correct scientific classification from the point of view of evolutionary history. The result of a molecular phylogenetic analysis is expressed in a phylogenetic tree. Phylogenetic inference is conducted using data from DNA sequencing. This is aligned to identify which sites are homologous. A substitution model describes what patterns are expected to be common or rare. Sophisticated computational inference is then used to generate one or more plausible trees. Some phylogenetic methods account for variation among sites and among tree branches. Different genes, e.g. hemoglobin versus cytochrome C, generally evolve at different rates. These rates are relatively constant over time, e.g., hemoglobin does not evolve at the same rate as cytochrome C, but hemoglobins from humans, mice, etc. do have comparable rates of evolution, although rapid evolution along one branch can indicate increased directional selection on that branch. A purifying selection causes functionally important regions to evolve more slowly, and amino acid substitutions involving similar amino acids occurs more often than dissimilar substitutions. 7. 5 stages of molecular phylogenetic analysis. Gene family evolution. Gene phylogeny is lines within Gray species phylogeny. Top, an ancestral gene duplication produces 2 paralogs, histone H1.1 and 1.2. A speciation event produces orthologs in the 2 daughter species, human and A speciation event produces orthologs in the 2 daughter species, human and chimpanzee. In a separate species, E. coli, a gene has a similar function, histone-like nucleoid structuring protein, but has a separate evolutionary origin and so is an analog. Gene duplication can produce multiple homologous proteins, paralogs, within the same species. Phylogenetic analysis of proteins has revealed how proteins evolve and change their structure and function over time. 9, 10. For example, ribonucleotide reductase, RNR, has evolved a multitude of structural and functional variants. Class 1 RNRs use a ferritin subunit and differ by the metal they use as cofactors. In Class 2 RNRs, the thiol radical is generated using an adenosylcobalamin cofactor and these enzymes do not require additional subunits, as opposed to Class 1 which do. In 3 RNRs, the thiol radical is generated using S-adenosylmethionine bound to a 4Fe4S cluster. That is, within a single family of proteins numerous structural and functional mechanisms can evolve. 11. In a proof-of-concept study, Bhattacharya and colleagues converted myoglobin, a non-enzymatic oxygen storage protein, into a highly efficient chempolyminase using only 3 mutations. This demonstrates that only few mutations are needed to radically change the function of a protein. 12. Directed evolution is the attempt to engineer proteins using methods inspired by molecular evolution. Molecular evolution at one site. One locus begins with a new mutation, which might become fixed due to some combination of natural selection, genetic drift, and gene conversion. Mutation. This hedgehog has no pigmentation due to a mutation. Mutations are permanent, transmissible changes to the genetic material, DNA or RNA, of a cell or virus. Mutations form errors in DNA replication during cell division and by exposure to radiation, chemicals, other environmental stressors, viruses, or transposable elements. If mutations to just one base pair of the DNA fall within a region coding for a protein, they are characterized by whether they are synonymous, do not change the amino acid sequence, or non-synonymous. Other types of mutations modify larger segments of DNA and can cause duplications, insertions, deletions, inversions, and translocations. 13. Distribution of rates for diverse kinds of mutations is called the mutation spectrum, CA. B of 14. Mutations of different types occur at widely varying rates. Point mutation rates for most organisms are very low, roughly 10-9 to 10-8 per site per generation, 15, though some viruses have higher mutation rates on the order of 10-6 per site per generation. 16. Mutations, a left-right arrow G or C left-right arrow T, are more common than transversions purine, adenine or guanine, left-right arrow pyrimidine, cytosine or thymine, or in RNA, uracil. Perhaps the most common type of mutation in humans is a change in the length of a short tandem repeat, e.g., the CAG repeats underlying various disease-associated mutations. Such STR mutations may occur at rates on the order of 10-3 per generation. 8 different frequencies of different types of mutations can play an important role in evolution via bias in the introduction of variation, arrival bias contributing to parallelism, trends, and differences in the navigability of adaptive landscapes. 19. 20. Mutation bias makes systematic or predictable contributions to parallel evolution. 40. Since the 1960s, genomic GC content has been thought to reflect mutational tendencies. 21. 22. Mutational biases also contribute to codon usage bias. 23. Though such hypotheses are often associated with neutrality, recent theoretical and empirical results have established that mutational tendencies can influence both neutral and adaptive evolution via bias in the introduction of variation, arrival bias. Selection. Selection can occur when an allele confers greater fitness, i.e. greater ability to survive or reproduce, on the average individual than carries it. Selectionist approach emphasizes e.g. that biases in codon usage are due at least in part to the ability of even weak selection to shape molecular evolution. 24. Selection can also operate at the gene level at the expense of organismal fitness, resulting in intragenomic conflict. This is because there can be a selective advantage for selfish genetic elements in spite of a host cost. Examples of such selfish elements include transposable elements, meiotic drivers, and selfish mitochondria. Selection can be detected using the Copper-kilosecond ratio, the McDonald-Kreitman test. Adaptive evolution is often found for genes involved in intragenomic conflict, sexual antagonistic coevolution, and the immune system. Genetic drift. Drift is the change of allele frequencies from one generation to the next due to stochastic effects of random sampling in finite populations. These effects can accumulate until a mutation becomes fixed in a population. For neutral mutations, the rate of fixation per generation is equal to the mutation rate per replication. A constant mutation rate thus produces a constant rate of change per generation molecular clock. Slightly deleterious mutations with a selection coefficient less than a threshold value of 1, the effective population size can also fix. Many genomic features have been ascribed to accumulation of nearly neutral detrimental mutations as a result of small effective population sizes. 25. With a smaller effective population size, a larger variety of mutations will behave as if they are neutral due to inefficiency of selection. Gene conversion. Gene conversion occurs during recombination, when nucleotide damage is repaired using an homologous genomic region as a template. It can be a biased process, i.e. one allele may have a higher probability of being the donor than the other in a gene conversion event. In particular, GCBIASED gene conversion tends to increase the GC content of genomes, particularly in regions with higher recombination rates. 26. There is also evidence for GC bias in the mismatch repair process. 27. It is thought that this may be an adaptation to the high rate of methylcytosine deamination which can lead to C-right pointing arrow T transitions. Dynamics of biased gene conversion resemble those of natural selection, in that a favored allele will tend to increase exponentially in frequency when rare. Genome architecture. Genome size. Genome size is influenced by the amount of repetitive DNA as well as number of genes in an organism. Organisms, such as most bacteria, Drosophila, and Arabidopsis have particularly compact genomes with little repetitive content or non-coding DNA. Other organisms, like mammals or maize, have large amounts of repetitive DNA, long introns, and substantial spacing between genes. The Z-value paradox refers to the lack of correlation between organism complexity and genome size. Explanations for the so-called paradox are twofold. First, repetitive genetic elements can comprise large portions of the genome for many organisms, thereby inflating DNA content of the haploid genome. Repetitive genetic elements are often descended from transposable elements. Secondly, the number of genes is not necessarily indicative of the number of developmental stages or tissue types in an organism. Few developmental stages or tissue types may have large numbers of genes that influence non-developmental phenotypes, inflating gene content relative to developmental gene families. Explanations for genome size suggest that when population sizes are small, many mutations become nearly neutral. Hence, in small populations repetitive content and other junk, DNA can accumulate without placing the organism at a competitive disadvantage. There is little evidence to suggest that genome size is under strong widespread selection in multicellular eukaryotes. Genome size, independent of gene content, correlates poorly with most physiological traits and many eukaryotes, including mammals, harbor very large amounts of repetitive DNA. However, birds likely have experienced strong selection for reduced genome size, in response to changing energetic needs for flight. Birds, unlike humans, produce nucleated red blood cells, and larger nuclei lead to lower levels of oxygen transport. Bird metabolism is far higher than that of mammals, due largely to flight, and oxygen needs are high. Hence, most birds have small, compact genomes with few repetitive elements. And evidence suggests that non-avian theropod dinosaur ancestors of modern birds 28 also had reduced genome sizes, consistent with endothermy and high energetic needs for running speed. Many bacteria have also experienced selection for small genome size, as time of replication and energy consumption are so tightly correlated with fitness. Chromosome number and organization. The ant Myrmecia pilosella has only a single pair of chromosomes, 29, whereas the adder's tongue fern Ophioglossum reticulatum has up to 1260 chromosomes. 30. The number of chromosomes in an organism's genome does not necessarily correlate with the amount of DNA in its genome. The genome-wide amount of recombination is directly controlled by the number of chromosomes, with one crossover per chromosome or per chromosome arm, depending on the species. Third, changes in chromosome number can play a key role in speciation, as differing chromosome numbers can serve as a barrier to reproduction in hybrids. Human chromosome 2 was created from a fusion of two chimpanzee chromosomes and still contains central telomeres as well as a vestigial second centromere. Polyploidy, especially allopolyploidy, which occurs often in plants, can also result in reproductive incompatibilities with parental species.